what we might do until she joins. So I was having a read before of your endosymbiosis theory questions, and I noticed, right, you're a bit confused on what to do. So what I've gone back and done is shown a proper question of what would they be asking you. Right, so an example, right, a very typical question for endosymbiosis theory. Explain the two structural features of a mitochondrion that support the theory that it originated from a free-living prokaryotic cell. Very typical question, nothing like unusual about it. So then, what do we kind of do to address the marks? And I've put it down as like a dot point process that you can kind of be ticking off. So the first thing, right, you need to find a structural feature that is unique to the prokaryotes.
Okay.
So you can see what we've identified here is the first structural feature unique to prokaryotes is going to be our circular DNA. Second point, you need to contrast that with what is the difference to its structural feature with eukaryotes. So you can see, I've just given some examples, right, what could be some of those differences. And then your final, like linking sentence, you need to explain that because of this difference, it suggests that the mitochondria are not originally part of the eukaryotic cell and that they are actually prokaryotic.
Okay.
So if we apply that to a scenario of what we've done before, right, you can see for our first sentence, mitochondria and chloroplasts have their own circular DNA. So that was marked 1 or dot point 1. I've identified the structural feature unique to mitochondria and chloroplasts. Mark number 2, I've identified it to or compared it to the fact that there's linear DNA in eukaryotic cells. And then to get mark number 3, I would write that hence this suggests mitochondria and chloroplasts were free-living prokaryotes. I think we did
Okay.
that in the second sentence. Yes. So you can see in this example, with that last line, I actually have that linking sentence suggesting they were engulfed via endocytosis.
Okay.
So what I might get you to do, let's pick the one for in between. So our prokaryotic and eukaryotic ribosomes. Following this structure of identify the prokaryotic structural feature, contrast it to the eukaryotic, and then explain why that would suggest it's a prokaryotic origin. So following that structure, can you write a paragraph suggesting why the ribosomal differences point to evidence of the endosymbiosis theory?
So is it like because mitochondria and chloroplast have a 7S ribosome?
Yeah. So that will be the first dot point.
So what should I start like the paragraph with?
So remember what we just said about that three-step pathway? Step number 1 is you need to identify the specific sequence or a specific feature for that prokaryote. So what would be for the ribosome, what's specific to the prokaryote?
It's 7S
It has a 70th. Now, why is that different to a eukaryote?
because eukaryotes have 80S.
Okay, so now we've established the difference.
Yeah.
What does that mean in terms of the endosymbiosis theory?
It means that uh mitochondria and chloroplasts came from prokaryotes.
So then what you just need to do, right, is state those three sentences together. So I'll screen share again. But what would you write as your answer to this? In this case, I only ask mitochondria, so you can specify that mitochondria contain their own 70S ribosomes
Oh, okay, yeah.
only found in prokaryotes.
And then do you say which are the characteristics of prokaryote?
So that will depend on the number of marks or what they've identified. So here they've identified two, right? So you need to talk
Oh,
about two
okay.
things unique to prokaryotes. So we could perhaps combine this with circular DNA as well,
Yeah.
plus circular DNA, only found in prokaryotes, whereas eukaryotes have ATS ribosomes and linear DNA. So that's the second dot point, right? We've contrasted it to what is the eukaryotic feature. And then
Okay.
the third, like wrapping up, you just have to link it back to the endosymbiosis theory. This supports the endosymbiosis theory that mitochondria were once free-living prokaryotes. Does that make sense?
Yeah.
Okay, so what I'll get you to do for homework then, we essentially did this one, but I'll carry forward the homework. So for the bottom
Okay.
two sentences, right, I'll get you to do it for homework this week. Follow
Okay.
this structure, right? So three-step process into how to differentiate the features.
Yeah. So is it the wait, so the home for the homework, which line is it?
So the two differences that eukaryotes reproduce via mitosis and meiosis, whereas our chloroplasts and mitochondria reproduce via binary fission. And then the second dot point is the fact that we have porins present in the membrane of mitochondria and chloroplasts, which are unique to prokaryotes.
Okay.
So this one and this one
Okay. Does it say prison? Like next to protein?
is porin, right? So it's spelled Porin. It's
What
a
does it mean?
specific type of protein.
Oh, okay.
So that's why I've written in brackets protein. It's some type of protein only found in prokaryotes, not found in eukaryotes, but we see it in the membrane of our mitochondria and chloroplasts.
Okay.
So these, like the ones I've given at the top, the ones for at the top are definitely the stronger evidences to go for, right?
Okay.
Because ribosomal differences, DNA, nucleic acid differences are bigger ones. Usually they'll only ask you to give two or three, so I would recommend you to choose from the top four. Just for completeness, I have given the last one as well, but it's not as big of a difference.
Okay.
Okay, so along the same lines, right, if we're doing questions, I'd like to give one more question as well as revision. So let's say we've got a cell specialized in secreting hormones. So hormones, remember, are a type of protein. Can you describe the organelles involved in this process. Now, before you go about answering the question, I want to talk about the steps of how do you know how much information to write. So when you get a question, what is the first thing you should be looking towards?
Like the keywords.
Even before we go to the keywords, there's something we need to check.
The topic or the
Not
marks?
the marks, right? So this question is going to be worth 4 marks. But it doesn't matter what the topic is, what the keywords are. If you don't know the number of marks, you never know how much information you need to be talking about.
Okay.
Once you know the number of marks, now you have to go back and look for the command terminologies. Right, so command terms is usually the starting word for every sentence, asking you to do or write about something. So in this case, the command term is going to be describe, right? That is the verb asking you to write an answer. Other command terms you will come across later is going to be things like justify, compare, explain.
What's the difference between like describe and explain? Or do they like mean the same thing, like in a question?
So each command term does have its own things, right? That's why we go through them.
Okay.
Describe is usually going to be requiring only one sentence, right,
Okay.
for 1 mark. Whereas when the command term is explain, right, it's similar, but it wants it in more depth, right? It wants more information. So you
Oh,
want to be
okay.
like 2 or 3 lines per mark,
Okay.
right? When we're talking about justify, that means it wants you to refer back to some sort of principle, right? So either refer back to key ideas, referring back to bioethics, something like that. You can't just write your ideas. You need to go back to biological definitions. That's what the command term justify means.
Oh,
And
okay.
the command term compare means it wants you to give similarities and differences. So those are probably the most common words.
Okay.
Actually, maybe one more, which would be sketch, for sort of diagrams or graphing, and that one is a bit more straightforward.
Okay.
So usually you'll have up to 2 command terms per question. Now, what you need to go back then is determine your mark split. Right. So if I have 2 command terms and a 4-mark question, how many marks am I going to allocate to each command term? We will do that in a few future questions because that's a bit harder to start off with. So as the first question, I've given you only one command term, which is the word describe. So you know all 4 marks are going to be attributed to the command term describe.
Okay.
Now, I said you should be looking to write 1 line per mark. So if it's a 4-mark question, you need to be looking at writing around 4 lines to get all the marks.
Okay.
So then, the command terms to the side. Now that you've looked at marks and command term, it's finally time for you to go and look at the topic. So what topic are we talking about here?
Organelles,
Organelles, good. Anything that can narrow it down more specifically?
like their processes
Which process?
of like the organelle, oh, of secreting hormones.
So remember, what did we say about hormones? They are a type of
Protein.
protein. So we're looking at protein secretion.
Okay.
Right. So what we're concerned about specifically for the organelles is all the organelles involved or helping with protein secretion. Now, if it's a 4-mark question, right, that means you are going to need to talk about 4 different organelles involved with protein secretion, and you need 1 sentence for each organelle to explain what is it doing.
Okay.
So if I start you off, right, let's say first organelle I'll talk about would be the ribosome. What is going to be the function of the ribosome? The ribosome,
Synthesized proteins?
so yes, remember that this one is secreting it. So is it going to be in the cytoplasm or the rough endoplasmic reticulum?
Wait, what does it mean by secrete?
Secrete means to leave the cell or to put it outside the cell.
Oh, so it would be in the Golgi, no, the endoplasmic reticulum.
So the ribosomes embedded in the rough endoplasmic reticulum are responsible for synthesizing synthesizing. Now, we can't just say synthesizing protein, right? This is a common mistake in bio. Why can't we say synthesizing protein? That's what we've learned so far in all of our theory. That's what it's responsible for. But the question hasn't specified protein. The question is an application, and it's specified hormones, which are a type of protein. So in your answer, you would now be expected to talk or replace the word protein with hormones everywhere,
Okay.
because they want to know only about hormones, not about all the other types of protein.
Okay.
So we'll say it's responsible for synthesizing hormones. Does that make sense?
Yeah.
Okay, so that would get you the first mark. Then what would be another organelle involved?
The Golgi,
What's the full name?
Golgi apparatus.
So we just want to specify the full terminology, right? Because Golgi can mean different things, whereas if you say Golgi apparatus, it's definitively referring to the organelle.
Okay.
Okay, and what would be its role?
To package the hormones and like further modify it and send it to the oh and like prepare it to be sent out of the cell in vesicles.
Yes. Which specific type of vesicle?
Exo some exocytosis.
Exocytosis is the process happening at the plasma membrane
Oh, okay.
for the protein to be released. We have 2 types of vesicles, right? So you've got the transport vesicle and the secretory vesicle. Which
So
one do we want here?
secretory.
Yeah, right. Ready for export. Okay, then what would be a third organelle involved?
Vesicle being an organelle.
Yes.
The vesicle, the excretory vesicles, transport it out of the cell.
So again, we need to be specific. You need to talk about start to finish, right? So where is it starting and where is it finishing?
Starts at the Golgi apparatus, and then it goes out to the plasma membrane to diffuse out of the cell. Or no, use a protein to go out of the cell.
So you used the process before. It started with E,
Cytosis.
exocytosis. Yes. So that's the process of excreting the protein or secreting the protein out of the cell.
Okay. But like, how does it happen with the protein? Or
So exocytosis, right, if this is the plasma membrane, this is your vesicle with the protein.
yeah,
It's a three-step process. So first step, it's going to move towards the plasma membrane from the Golgi apparatus. Step number two, it fuses, right, with the plasma membrane.
okay,
Now, once it's fused with that plasma membrane, right, this layer here can disappear. And so it's got a fusion point here and a fusion point here.
okay,
Step number three then is that this is pushed right upwards to reform that circular structure of the plasma membrane because cells usually are circle or oval shaped.
okay,
And as it gets pushed upwards, right, that protein also gets pushed out of the cell then.
okay,
So that is what we call exocytosis.
okay,
Right. So your key word that you need to use is step number two, which is where you have to say it fuses with the plasma membrane.
okay.
Now, you wouldn't need to talk about it in this much depth for this question here because they just want one sentence. So you can just say exocytosis happens. If it was a 2 mark, right, assigned to one organelle, right, that's when you would need to talk about exocytosis in depth. So
Okay.
that's why we're looking at the number of marks, because 1 mark means you can just say exocytosis. 2 marks for vesicles would mean you now need to explain what is exocytosis and how does it happen.
Okay.
Okay, so we've got three organelles then. We need one more organelle involved in the production of a protein.
Um, maybe the rough endoplastic reticulum.
Yeah. And what does it do?
It synthesizes the hormones and prepares it to be sent out to the Golgi.
Hang on. We said the ribosome is responsible for synthesis.
Oh yeah, yeah. I meant the ribosomes on the rough endoplastic reticulum. Do that.
So the ribosomes are, yes, embedded in the rough endoplasmic reticulum, but that's not going to be the function of the rough endoplasmic reticulum, right? That's just something else that's there by coincidence.
Okay.
What
Um,
actually happens to a hormone in the rough endoplasmic reticulum?
it modifies it.
Yes. So the Golgi apparatus does the bulk of modifications, but rough endoplasmic reticulum can do a little bit. But there's one key thing we're searching for.
Oh, folding.
Yes, folding. Right? At the ribosome, you just get a straight line, right? It's just synthesizing that protein. It's the rough endoplasmic reticulum that will fold the ribosome or the protein into a shape or correct shape.
Yeah.
And the Golgi apparatus does final modification. You can also say that it will package it into a transport vesicle to take to the Golgi apparatus. That was also folding and packaging is again two key roles of the rough ER.
Yeah.
So that would be how like your model response should look like for a 4 out of 4 answer.
Okay.
And I've also typed it out. So just if you want to have a read,
Where is it? Oh, here.
I
Yeah.
was just adding it in. So you can see you'll get 1 mark for each organelle. Now, usually the three we say that are most important is going to be Raphia, ribosomes, and Golgi apparatus. So you probably do want to be talking about those three at minimum. Now,
Yeah.
given it was 4 marks, you have a couple of extra options to talk about. So you could pick a vesicle to talk about the transport. If you wanted to explain exocytosis in depth, you could talk about the plasma membrane, or you could talk about mitochondria, which is providing energy for it.
Oh, okay.
Yes. So, but those three, right, you have to pick one of these three, but you definitely talk about all these three because they're the main ones involved.
Okay.
Does that make sense? Any questions?
Yeah, it makes sense.
Yeah. Perfect. Hi Jade, are you there?
Yeah.
Yeah, perfect. So I didn't notice when you joined. Can you quickly tell me how much have you listened to?
The past like uh 5 minutes.
It's okay. So what we've kind of just done is we've talked a little bit about the homework, when we were talking about writing a model answer to the endosymbiosis theory question, and we've also addressed how to answer some more 4 mark questions. I won't go through it again, but it might be a good idea for you to watch the first 20 minutes on recording or so that you can understand how we went about answering these two questions. Okay, so we'll go on with the content now. So every class, first class we did purely content, but every class from now on, we're going to try to split some content and some question practice as well in every single class. So we'll start with the content now. Last week, we finished the topic of the plasma membrane. So this week, we're going to be starting the topic now of transporting molecules across the plasma membrane. Right. The broad topic name is called transport, right? Or diffusion. Now, transport is split into a particular subcategories. So I'm just going to split that for you as well. You have passive transport and you have active transport. Does anyone want to guess the difference between passive and active?
Active requires energy and passive just like doesn't require because it's like passive.
Yeah, perfect. So it's a pretty intuitive explanation. Now, for biology, right, we want to try to be using key terminology. So instead of saying requiring energy, what I want you to be focusing on is say requires ATP, right? Because that is the energy currency of the cell. So I'll write that down. Requires ATP. Okay. Now, passive transport is then further split into two types again. So that will be split into osmosis, which is for water, and diffusion for everything else. Active transport, same thing, is split into two different types. That is bulk transport as well as active transport. So we'll be starting with the passive processes as they're a little bit easier. So firstly, we'll talk about diffusion. Now, if you think about what diffusion is, right, it's the spread of molecules across the room, right? Think about the fact that if you've got a cup of water and you still that cup, right, the water's just not going to sit there at the bottom of your desk, right? It's going to spread across your whole desk. That process of it spreading out across your whole desk, that's diffusion happening right there. So what's the definition of diffusion? Diffusion is the movement of particles down their concentration gradient, right? So this is a really key terminology to remember. Whenever they ask you to define diffusion, you want to be saying down the concentration gradient or from an area of high concentration to low concentration. But I think saying down the concentration gradient is easier than that. So if you don't just take our water example, right? When you spilled that water, initially that puddle, right, it has a very high concentration of water, and the rest of your desk doesn't have any water in it. So it's a very low concentration. So that's why your water is going to spread out across the entire desk because it's moving from high concentration to low concentration, or it's moving down its concentration gradient. Now, when we're talking about diffusion, we'll usually be talking about it in the context of our plasma membrane. So if you remember what we said last week, plasma membrane is semi-permeable or it is selectively permeable. So it chooses what things can cross it and what molecules cannot cross it. For diffusion to work, the molecule must be able to cross the plasma membrane. Right, so when you're thinking of oxygen that can cross the plasma membrane, it could undergo diffusion. If you're thinking of Na sodium plus, right, Na plus, this is a charged ion. It cannot cross the plasma membrane, so it will not undergo diffusion inside your cells. Does that make sense so far?
Yeah.
Okay. So as a quick recap, I might just get you to list the properties of molecules that can cross the plasma membrane. So I'll start you guys off. One of the properties that a molecule needs to cross the plasma membrane is that it should be a non-polar molecule. So Liana, can you give us another property?
It has to be small in size.
Yeah. And Jade, what's another property?
Is one of them like it has to be like hydrophobic?
It's hydrophobic. And then the very last property is it should be uncharged. Right, so these four properties should be present or they should be attributed to a molecule for it to cross the plasma membrane. And only when that is possible can we talk about that molecule doing diffusion. So let's give a diagram. Actually, it might be better if we draw it like this. So imagine this is your plasma membrane. You've got your fatty acid tails, your phosphate heads, right? Some channel proteins, cholesterol, peripheral proteins, etc., etc. So let's say you've got oxygen in blue on one side. This is probably representing your bloodstream, right? Because it's O2 rich, right? There's lots of oxygen here. Now, in contrast, right, you've got a bunch of cells here. These cells, they need oxygen to survive, right? You need oxygen to breathe. You're using oxygen to breathe. So they're O2 poor, or there's a low concentration of oxygen. So what's going to happen is that oxygen is going to diffuse across that plasma membrane. And let's take one specific cell, intracellular space. It will move across, right, because it's small, it's non-polar, it's uncharged, and it's hydrophobic. And we end up with lots of oxygen inside the cell. So that's the process of diffusion. If the cell, let's say, wasn't using any oxygen at all, what should happen after a couple of hours is that the amount of oxygen inside and outside should be exactly the same. So we've got 6 oxygen outside and 6 oxygen inside the cell. It's completely equal. However, because you're using oxygen to breathe, right, you're effectively always eliminating oxygen inside the cell, and your bloodstream is always bringing in new oxygen. So you're always getting diffusion going down for oxygen. It's always diffusing out of the bloodstream and into your intracellular space. If we use a different example though, so let's say we've got a beaker here. You've got a semi-permeable barrier here. You've filled it with water and you've got some molecules permeable here. So what will happen over time, right, is that these molecules move across randomly and eventually, right, because they don't like colliding with each other, they want to be spaced apart as far as possible. You end up with 5 molecules on this side exactly. Now, at this point, we say there is no net diffusion. Once there are 5 molecules on both sides, but you cannot say that diffusion stops. But still, this molecule will move across to this side, and this molecule will move across to this side, right? The molecules continue moving around randomly. And so diffusion is still occurring, but the molecules are still moving. There's just no net diffusion, right? That 5 molecules will remain on either side. It won't suddenly go 6, 4, or 7, 3. But the 2 individual molecules, they can still swap around. So you need to be careful that you don't imply that movement stops, right? And to say that, you can use the terminology that there is no net diffusion occurring once you've reached that point. Now, the other thing you need to know, if we talk back to our oxygen example, the greater the difference in concentration, the faster the rate of diffusion. Right, so if there is just one, let's say 4 oxygen molecules on this side, on the bloodstream, and 0 in the intracellular space, you'll have diffusion occurring, yes, but that diffusion won't be very fast. Whereas if you've got a bunch of oxygen on one side, now that diffusion process is going to be very, very fast because they'll collide with each other more and they'll want to separate themselves out quicker. So the scale or the difference in the concentration gradient is a really big determiner of the rate of diffusion. I'm just going to eliminate that. Rate of diffusion depends on concentration gradient. It also depends on one more thing, which is the surface area. Right, so even if you had a really, really big difference, actually, let me redraw that. right? So you've got so many molecules here. So this is a really big concentration difference, but the surface area for diffusion to occur is quite small. So it would be difficult for diffusion to occur quickly in this case because there's not a lot of space for the molecules to move across. In contrast, if you put that same container on the bottom, now there's a lot of surface area for diffusion to occur, and diffusion would occur here very, very quickly. Now, one point to remember, let's redraw this diagram. Diffusion is dependent on concentration, not on amount. So if you assume this represents 2 units and this represents 1 unit length, if you are starting with 12 molecules in the upper box, after some time, right, you do not end up with 6, right? You only end up with 6 if they were the same space, they had the same volume. You would, in this case, end up with 8 molecules in the upper region and 4 molecules in the lower region. Why? Because that represents the same concentration. Right? In this example, there's 4 molecules per unit space in both boxes. And concentration is your number divided by your volume or your total space. So if one side has more space than the other, it will have more molecules in that region proportionate to its space. So if you stay with the same example, and now you put this to be 3 units, right? So it's 3 times as big as the smaller box beneath. You are going to have 1 more molecule here move to the top. And that will now represent 3 molecules per unit space, right? The bottom one is 1 unit, so it has 3. The top one is 3, so 3 times 3, it has a total of 9 molecules in it. Does that make sense?
Yeah.
Any questions? Okay. So, what I want to quickly cover then is some of the common molecules that you should know that can do this sort of process, right? So they can just diffuse across the plasma membrane. We've covered the example of oxygen quite a few times, so hopefully you remember that now. The other important one to note is carbon dioxide. Right, so most of our gases, like oxygen, carbon dioxide, nitrogen, they don't have a charge. They're really, really small. And so they're all able to diffuse across the plasma membrane. Now, nitrogen is not very important, so we don't tend to talk about it. Oxygen and carbon dioxide are really, really important. So those are 2 gases that you should remember can cross the plasma membrane. The other one is our hormones. Right, so those are ones present in the bloodstream. I think we talked about it earlier in one question. Right, and they are very, very hydrophobic. Right, they hate water. And you remember that the plasma membrane's interior, composed of those fatty acid tails, is also hydrophobic. It hates water. So the 2 mix well together. And lastly, interesting molecule actually, but our alcohols, right, especially ethanol, that's the molecule we talk about in biology, right, it is also able to cross the plasma membrane. Right, why? Again, it's a small molecule, it's non-polar, it doesn't have a charge, so it's able to cross. So these are some good molecules to remember. Right, either they're simple or because we'll cover them in a different context, and so you can kind of double up by remembering it already. Okay, so that's simple diffusion, right, straight through the plasma membrane. We also have facilitated diffusion. Facilitated diffusion is using the proteins that we talked about last class, right? So our protein channels are particularly, or you also have something called a carrier protein, which I'll talk about in a sec. Right, but you have to use one of those two transmembrane proteins to allow the molecule to cross through the plasma membrane. So firstly, why does it need this? It's usually because the molecule is really big or it is charged. So example, glucose, right? The molecule responsible for your whole body's energy, it's a really big molecule, right? If it has to supply so much energy, it has to be a big molecule. So it can't just cross the plasma membrane. It needs help to do that. So it needs proteins help. Or you can think about our favorite example, sodium. Why couldn't sodium cross? It's because it was a charged molecule. And if it's charged, it can't cross. So these two molecules, they're going to need help to cross the plasma membrane. The process of proteins helping them cross, that's what we call facilitated diffusion. The proteins are facilitating the diffusion to happen. So let's talk about our facilitated diffusion. Two types, channel protein, carrier protein. When we're talking about the channel protein, this is the simpler example. It's just a big protein, has to be transmembrane protein, and what it has is a open channel. Right, so this open gap straight through the center of the protein. What this allows, particularly, is things like ions, Na+, K+, Cl-, Ca2+. So those are some of our common ions we're talking about to travel through the gap in the channel protein and into the other side of the plasma membrane. Why is that possible? Well, remember the protein is occupying this entire space. So suddenly there are no phospholipids here. And so there is no hydrophobic or water-hating environment. The protein creates its own environment, which allows charged molecules, especially ions like sodium, to pass through. So you can quite literally think of it as a hole in the plasma membrane to allow particular ions to flow through. Now, just please note that these channels, they are specific, right? So you will have a specific sodium ion channel protein. Right, now potassium can't come and use this. Right, potassium would need its own potassium ion channel protein. And then calcium couldn't come and use this. Calcium would need its own. Right, so for every molecule, there will be a unique channel protein to it. The other type of molecule you have is a carrier protein. These ones are slightly more complex, and they're usually used for larger molecules. Right, so a good example for this is glucose. So you can see we've got a high concentration of glucose on this side and a lower concentration of glucose on this side. So glucose wants to move through, right? It wants to diffuse to the other side, but it's not able to do that because it's too large to cross the plasma membrane. So it needs help. It needs the help of carrier proteins. Now, how do carrier proteins help? It's a three-step process. So what's going to happen? Step number 1, our carrier protein is looking something like this-ish. It might be easier if I actually shade it in. So what it has is a gap on this side. Let's draw in our molecules for glucose again. Now, a molecule for glucose can now come in and bind to the center of that protein. Glucose molecule binds. Now, this is a really key terminology you need to remember. This triggers a conformational change in shape. So change in shape is obvious, right? The protein now will be changing shape or looking different. What does conformational mean? Conformational means it's in response to some sort of event. In this case, the event is glucose binding to the center of the protein. You can't forget the word conformational, okay? So you need to write the whole thing, conformational change in shape. Right, that is proper bioterminology. So what that change in shape results in is our protein closing the top and opening the bottom. Right, so we've got our glucose molecules here, but the glucose molecule that was inside the protein, now the top is sealed off, it's shut off, and the bottom opens. And so our glucose molecule can continue its facilitated diffusion down to the other side. And finally, step number 3 is reset. Right, so the molecule will go back to its original shape. Right, where it is open at the top and closed at the bottom. And then it can restart the entire process. So that is how our carrier proteins are working. Right, and your key word, which I've highlighted in blue, is to use conformational change in shape to describe this process occurring. Now, just like the channel protein was specific, right? So you would have a sodium channel protein, you would have a potassium channel protein. Carrier proteins are specific too. Right, so glucose will have its specific carrier protein, and then it can't go use the carrier protein for triglycerides. Those will have their own specific carrier proteins. So just some key dot points for you guys to note. I don't think it's combined, right? Yes. Passive, right, means it's not going to require ATP. Why? Because it's moving down the gradient. Everything is specific for channel and carrier proteins. They only work for one molecule. Conformational change is your key word you need to remember for carrier proteins. Your channel proteins are often going to be used for ions, particularly sodium. And your last one, right, this process is very, very efficient, right, especially for channel proteins, because it's going to allow a continuous flow. So those are just some key things to remember. Now, in terms of question preparation, right, if you start to see a graph that looks like it's leveling off, or the correct biological terminology is plateauing, right, as the concentration increases. So what I mean by that, if this is representing diffusion rate, and this is representing concentration gradient, if as the concentration gradient increases, your diffusion rate starts to level off, right? It starts to plateau, remain at the same level. This is usually an example of facilitated diffusion. Right? Because your proteins work in here, right? Your carrier proteins specifically, right? They become saturated or they're working at their maximum capacity, right? They're going from this form into step number 2, where they're flipped around, into step number 3, reset, as fast as they possibly can. Right. You usually wouldn't have this for diffusion because remember diffusion could occur across the entire plasma membrane. So it's really unlikely that you run out of space for diffusion to occur because the plasma membrane is just so big. But the proteins, right, the channel proteins and carrier proteins, you don't have as many of them. And so if you start to run out, if you start to plateau, it's probably because it's an example of facilitated diffusion and you not having enough carrier proteins or channel proteins to continue increasing the diffusion rate as concentration gradient increases. Does that make sense?
Yeah.
Any questions? Yeah. Okay. So we'll talk about osmosis as well. Oh, we need to talk about, what affects the rate of diffusion quickly. We did already talk about some of the big factors, right? So main one is going to be concentration gradient difference. Concentration gradient and surface area. Can you guys think of any other factors that would affect the rate of diffusion?
The size,
Size of?
molecule,
Yes. So it's easier for smaller molecules to diffuse through the plasma membrane, and it's harder for larger molecules to diffuse through, right? If you think of a jungle, right, really closely packed trees, it's a lot easier for people who are shorter, who are smaller, to weave their way through the jungle than for someone who is a 6 foot tall, fully grown adult. So smaller molecules have it easier. What else could affect the rate of diffusion? Jay, do you want to give an example or brainstorm an example?
Um, I can't really think of anything other than like those three.
That's okay. Liana, do you have anything else you can think of?
no, not really.
Okay, so if we talk about some of the other things, right, remember what we talked about with the plasma membrane? What caused molecules to become really excited and move quickly? That was temperature. Right, so at higher temperatures, your molecules will have more kinetic energy and they will move quicker as a result. So the example we talked about that I think was water or liquid water, ice, and water vapor. Right, at really low temperatures, your water becomes a solid. It doesn't move from its shape. At room temperature, it's a liquid, right? So it starts to spread a little bit, but at high temperatures, it's going to be a gas, right? And it will quickly spread and move across the whole room really, really quickly. Why? Because there's more kinetic energy, there's more movement energy. Same way, diffusion is just a process of random movement, and over time, balancing the random movement on both sides of a semi-permeable barrier. So if you have quicker random movement, right, you will hit diffusion or you'll hit net diffusion faster. So higher temperatures will increase diffusion rate, lower temperatures will decrease diffusion rate. These are probably the best four to go for. But just for completeness, I would mention the other ones, but they're not as great of factors. We have the width of the medium or the width of your semi-permeable barrier. So remember how we said you could have a phospholipid bilayer or you could have a phospholipid unilayer. Right? You could also have a phospholipid trilayer or a quad layer, but it just depends on how many layers you have. Now, the more layers you have, the harder it is for diffusion to occur. The less layers you have, easier it is for diffusion to occur. But if we continue with the analogy, think of a unilayer as you having to just run through one line of trees, whereas if for a bilayer, you're having to run through a whole forest, right? Multiple layers of trees. It's a lot easier for you to run through a single layer than for there to be multiple layers. So it's easier when there is less layers or the width is smaller. Also, permeability of the membrane. So we did a lot of talking about the importance of cholesterol, right? As regulating the fluidity of the membrane. And we also talked about saturated versus unsaturated fatty acid tails. Would anyone like to quickly volunteer and explain what was the difference between saturated and unsaturated fatty acid tails? That's okay, I'll go back to it. So our saturated, no, this is too far off. It was on this one.
Was saturated like only one? And
So saturated means there are no double bonds.
oh, okay.
Yes. Whereas unsaturated means there is a double bond. So if it is saturated, right, your tails are completely straight. Whereas if it is unsaturated, your tails are bent, or we say they have a kink. Right, and that's because of the double bond. When you have a double bond, you can't join it in a straight line. It has to join off in an angle. So it's caused by being that double bond.
Yeah.
Now, if you're trying to stack things together, right, it's easy to stack them when they're straight and flat. It's hard to stack them if they're bent at that awkward angle. So if our phospholipid bilayer has only saturated fatty acid tails, right, what happens is that those fatty acid tails are stacked really, really tight together. That makes it very difficult for diffusion to occur because there's not much room or space or gaps for the molecules to go through. Whereas if we're talking about unsaturated fatty acid tails, right, suddenly, I should draw this to the side. Right, there's a lot of room for diffusion to occur because the molecules are spread further apart. Right, so I hope you see how those two topics link together. Saturated or unsaturated fatty acid tails determine the amount of membrane permeability, which means it influences the rate of diffusion. Uh, we're a bit too far down. Here, rate of diffusion. So those are some of the other ones. I have written them all down in words again, but just wanted you to think about it on the spot. What could be some of the factors? These four are the best ones to go for, but do keep these in mind. Okay, so about this diagram I was talking about before, have a look here. So we've talked about two rates of diffusion, right? Facilitated diffusion and simple diffusion. Can we explain which methods of diffusion the dotted blue and solid red lines represent and explain why they follow that trend? So I'll give you 3 minutes for 3 marks. Can we try to write an answer to this question?
Um, I'm kind of confused about this question.
Uh, what specifically are you confused on?
Well, I understand how to like read the graph, but I'm like not entirely sure like which one's which.
So, as a hint, think back to the tip we just talked about earlier, right? When something tends to level off, it's because there's a limited number of options for the molecule to diffuse through. So, usually, if we're talking about facilitated diffusion, there's a limited number of channel and carrier proteins. So if you continue to increase the concentration gradient, but you don't increase the number of channel or carrier proteins, you can't increase the rate of diffusion further. Whereas,
Oh, okay, I get it.
yeah, the plasma membrane is really big, so you don't have a limited number of it. You've got unlimited.
Hello.
Yeah.
Wait, what are we doing?
So once you've written your response, can you send it to me?
Oh, okay. Oh, I thought that. Oh, never mind.
Did you do it by hand? Okay. So I'll go through it. Your red line is representing facilitated diffusion. Your blue line is representing simple diffusion. How do we know that difference? Because as we just explained, facilitated diffusion should put 2 off because your carrier proteins and your channel proteins are limited. So if you have a read of this response, right, that's what I'm explaining here. Your red line represents facilitated diffusion. Why? Because there's a limited number of channel or carrier proteins. As concentration gradient increases, they become saturated, right? Or they're working at maximum possible capacity. Now, your blue line represents simple diffusion, right? And there is no real limit for simple diffusion because across the phospholipid bilayer, right, it's got enough surface area to keep increasing diffusion rate as concentration gradient increases. So for your mark allocation, where are the marks going to be? You'll get 1 mark for explaining red line is facilitated and blue line is simple. You'll get 1 mark then for your explanation of why this is facilitated and 1 mark for your explanation of why this is simple diffusion. Right, so Liana, your response, right, is perfect. Right, so you've identified red line is facilitated and blue line is simple, and you've explained then why is each why is it simple and why is it facilitated. If you want, you can send me your response like once we're done, Jade, or outside of class, and I'll mark them as well. But we'll make a move on for now. So the next topic is going to be osmosis. Now, let's just quickly go back up. Let's cover this earlier. But as a quick recap, right, we should know what these 3 terms mean. The solvent is the liquid that is dissolving something or some molecule. Now, for biology, this is always going to be water. Right, so your solvent will be water. Your solute is what gets dissolved in water. And your solution is the combination of solute plus solvent. Okay, so far for diffusion and facilitated diffusion, you've been focused on the movement of the solute. For osmosis, we are going to focus on the movement of the solvent. Right, so we're looking at a different thing. We're looking at the movement of water now, not what is dissolved in the water. So whenever we're talking about movement of water, just note then the terminology is osmosis. Do not use the term diffusion to talk about movement of water. Okay. So what is osmosis being used? Osmosis is going to be used when you have a semi-permeable barrier that the dissolved molecule cannot cross through, or the solute cannot cross through it. So as an example, let's say you've got a beaker here. You have equal amounts of water on both sides of the beaker, and let's say yellow represents urea, right, which is a solute. Urea is charged, right? It has a positive charge. So it cannot cross this barrier if you imagine that's the phospholipid bilayer. Why is that a problem? Well, if you have 6 urea molecules and 2 urea molecules on this side. What urea wants to do, right, is move across to this side so that you end up with 4 urea molecules on both sides. And now everyone is happy because you've got the same concentration on both sides. So this is what we have been exploring so far, which is diffusion. However, if the membrane is not permeable, right, so this cannot cross the membrane because it's charged. What happens instead is water has to cross the other way. Because ultimately, our goal is to get the same concentration. So if we cannot put urea to the right side, what we'll do is we'll take water to the left side. And what you end up with is a really high water level on one side and a low water level on the other side. Specifically, the water level on this side will be 3 times the height of the right side because there's 3 times as many urea molecules and we need to maintain the same concentration. So this is called osmosis now because we're looking at the movement of water instead of the movement of the solute, right? So that's what's changed. Now, how osmosis works usually, or the definition I should say, is water moves from an area of low solute concentration to high solute concentration. So what's solute? Remember, solute is the thing dissolved in the water. In this case, that's urea, those yellow circles. So the water we see, it's moving towards the left side because there are more urea molecules there. We have the same amount of water per urea molecule. So why would the cell do this? It's because urea is a very difficult molecule to get across the plasma membrane, right? It's large, it's charged. So it's easier for the cell to move water across instead. Now, if you remember, water is also a polar molecule, so it can't diffuse across the plasma membrane. However, we do have something called an aquaporin, right, which is a channel protein for water, aka water has to do facilitated diffusion across the plasma membrane. Facilitated diffusion. So that's what is happening here. Now, what I've explained is simple terminology. Unfortunately, you need to use the proper terminology when describing this process. So what are those proper terminologies? We don't say high solute or low solute concentrations. We use the terms hypertonic, isotonic, or hypotonic. So let's start with hypertonic. Right? Hyper means more. Tonic is referring to tonicity. Tonicity is how we compare solutions, or to be more specific, compare solute concentrations. In the solution. So if we say the solution is hypertonic, right, that means the solute concentration is higher outside the cell. So just to paint the diagram of what's happening, this is your cell. This is outside the cell. We are talking about the fluid here, right? The fluid outside the cell. So we say that fluid is hypertonic. It means it has more solutes than the cell, and the water will move from the cell outside the cell. When we say the solution is isotonic, that means there is the same solute concentration inside and outside of the cell. Now, remember what I said. Your key terminology is going to be no net movement. You must use the word net because there is still movement, right? So you can't say there's no movement. It's just that the movement into the cell equals the movement out of the cell, right? So the official terminology for that is something called dynamic equilibrium, but it's basically just a fancy way of saying that you have equal amounts of water entering the cell and equal amounts of water leaving the cell. Right, so there is no net change in water. And finally, the last word to describe the fluid here outside the cell is going to be hypotonic, which means the solute concentration is less outside the cell compared to inside the cell. And that means water is going to be moving into the cell, right? So the cell will swell or burst because water is moving into it. Does that make sense to everyone? Any questions?
No.
Okay. So, oops, I'll just write that again. So, as a quick example, right, so let's say here is our cell. The cell has a concentration of 0.5 molar sodium chloride. Outside the cell, the concentration is 0.2 molar sodium chloride. Now remember, when you're using the terminologies, you are comparing the concentration inside the cell to the concentration outside the cell. So in this case, is the solution inside the cell hypertonic, isotonic, or hypotonic?
Hypotonic?
Sorry, can you repeat that?
Hypotonic?
So the cell's concentration in this case is higher than the concentration outside.
Wait, is the question like, is water going into the cell?
So what will happen, right, is the question will always be in two parts. I'll just quickly mention it. You always first be asked to use some tonic terminology, and then you will be asked about water direction movement. So I'll maybe just go through this example. Right here, I would describe the solution inside the cell as being hypertonic because it has a higher concentration than the solution outside the cell.
So the salt inside the cell is more than the salt outside the cell?
The solute concentration, yes.
Okay.
Now, just so you know, you can define it in either direction. So the question I specifically asked was compare the cells concentration to the fluid surrounding the cell. They can also ask you, compare the fluid surrounding the cell to the cell's concentration. So the cell is hypertonic and the fluid outside is hypotonic. Does that make sense? You might be asked to describe either one, right? You might be asked to describe this fluid or this fluid. So you need to pay attention to which one they are asking you to describe. Now, because the concentration of solutes is greater inside the cell than outside the cell, that means water is going to move into the cell so that we can try to get equal concentration. So if I pose an example to you then, I'll go to the right. Here is outside the cell. Here is the cell. The cell is 0.3 molar. Outside the cell, NaCl. Outside the cell, we have 0.7 molar NaCl, extracellular. So, number one, describe the tonicity inside the cell. Right, so we're talking about this solution. Is it hypertonic, hypotonic, or isotonic?
Be high hypertonic because there's the water wants to go outside of like the water goes out of the cell.
So the water, remember, is going to move from high concentration to low concentration.
Like is it a high concentration of water or high concentration of salt?
High concentration of the salt.
So it's gonna go in. Like it's gonna be hypotonic because it's gonna go into the cell.
Which does outside the cell or inside the cell have higher concentration?
Outside.
Outside. So if water is moving to higher concentration, will it be going outside or inside the cell?
I thought it's from a high concentration to a low concentration.
So that is diffusion, right? This is what I talked about here. Diffusion, which is what we talked about at the start of the class, is high concentration to low concentration. But osmosis is different. Osmosis is looking at water, and water moves from low concentration of solutes to high concentration.
Solvent.
Still solute.
Okay. Wait,
Right,
what
so
would the solvent one be then? Would it be low to high solvent concentration of solvent as well?
so we say water doesn't have a concentration, right? You can't really say I have 1 liter water per 1 liter water.
Yeah,
So
okay.
we only use concentration when we're talking about solutes.
Okay.
Now, if you are talking about the solutes switching sites, so you have a semi-permeable barrier, you are talking about diffusion and diffusion happens from high concentration to low concentration.
Okay.
If you are talking about osmosis, so you have an impermeable barrier and water is switching sides, then you are going from low concentration to high concentration. So
Okay.
there's two things happening, and what you need to determine is, am I looking at diffusion or am I looking at osmosis? Now, in this question, right, because we are talking about water moving, we are talking about osmosis.
Okay.
For osmosis, right, water will move from an area of low concentration to an area of high concentration.
Yeah.
So water moves
So it's
out
hypertonic
of the cell. Which solution is hypertonic? Inside the cell or outside the cell?
inside because it's losing water. It becomes shriveled.
So what to remember, right? I think easy way to compare it, just look at the numbers, right? The one that has the bigger number is going to be hyper, and the one that has the lower or smaller number will be hypo.
Okay.
So what does tonic refer to? Tonic